Hello, this is me Iramgul and you're watching my YouTube channel Chemtracks by IGM. Today I am going to tell you about protein structure and conformation. Basically, the special arrangement of atoms in a protein is called its conformation. The possible conformations of a protein include any structural state that can be achieved without breaking covalent bonds. A change in conformation could occur, for example, by rotation about single bonds. Of the numerous conformations that are theoretically possible in a protein containing hundreds of peptide bonds, one or a few generally predominate under biological conditions. The need for multiple stable conformation reflects the changes that must occur in most protein as they bind to the other molecules or catalyze different chemical reactions. The conformations existing under a given set of conditions are usually the ones that are thermodynamically the most stable, having the lowest Gibbs free energy. Proteins in any of their functional folded conformations are called native proteins. There are different levels of structures in proteins. First is primary structure. You can see this is basically linear arrangement of amino acids is known as primary structure. Then there are some helical or sheet-like structures or composed secondary structure of proteins, and they contain alpha helix and beta plated sheets. Tertiary structure and quaternary structures are basically most complicated, or they are basically folded structures of protein. So let's discuss one by one. Basically the simple amino acid sequence of protein is called as its primary structure. This is basically the simple or straight chain sequence of amino acids. These small balls are basically amino acid units or amino acid molecules like phenylalanine, leucine, serine, and cysteine. Since the possible way of arrangement of the chain will depend on the sequence of amino acid residue leading to the proper protein folding, the primary structure dictates three-dimensional structure of protein. Although it is a linear sequence, or it is a simple amino acid chain sequence, but uh, it it forms the three-dimensional structure of protein. Next is secondary structure. This defines the interaction of closely located amino acids in a chain. Two main type of secondary structures observed in the proteins are helix and pleated sheets. Alpha helix is a helical structure around an axis. This is coiled in clockwise manner. It has an average of 3.6 amino acids per turn. There are 3.6 residue per turn with the fixed pitch of 5.4 angstrom. Thus, the rise per residue come out to be 1.5 angstrom. Basically, as I told you earlier, a secondary structure is consist of pleated sheets and helix. This is basically helical structure or alpha helix, and this is beta plated sheet. Alpha helix, you can see this one is basically alpha helix, and rise pattern is 5.4 angstrom. The reason as to why alpha helix form more readily in proteins than any other possible conformation is that these arrangements make optimal use of internal hydrogen bonds to attain stability. You can see here some dotted lines. These are basically hydrogen bonds between any of the electronegative atom, either it is oxygen or nitrogen, and it can form a hydrogen bond with the hydrogen atom of other amino acid residue. The helix is stabilized by the hydrogen bonding between the carbonyl of each first amino acid of the chain to the N-nature amino of the amino acid 4 residue per away. So it is basically hydrogen bonding between 1 and 4 amino acid residue. All main chain amino acid and carboxylic groups are thus hydrogen bonded, and R groups are stick out from the structure in a spiral arrangement like this one. This is R group or side chain of the amino acid. Beta pleated sheet is composed of two or more straight chains that are hydrogen bonded side by side. If the amino acid termini are on the same end of the each chain, the sheet is termed as parallel. But if the chains run in the opposite direction, amino acid terminal on opposite end, the sheet is termed as anti-parallel. Pleated sheet may be formed from a single chain if it contains a beta turn, which forms a hairpin loop structure. Often, a proline can be found in a beta turn since it places in a kink in the chain. Glycine and alanine are predominant amino acid in beta sheet. This is the structure of the pleated, and here is the amino arrangement beta plate sheet. Next is the tertiary structure. Tertiary structures refers to the arrangement of amino acid in a space like is the three-dimensional form. Distinct amino acid are brought closer in a chain or further linked by different interactions or different forces of attraction like polar-polar interaction, hydrophobic attractions or interactions, ionic disulfide bonds, van der Waal bonds, and hydrogen bonds. Hydrophobic amino acids are buried inside the core of protein, and charged and polar groups are located on the surface, which tend to closer and exclude water. This arrangement basically allows a protein to have great water solubility. The higher one is quaternary structure, and if protein consists of more than one polypeptide chain, their association with each other implies a quaternary structure. For example, this is a quaternary structure, and there are you can observe two different colors. These purple and light purple colors are basically showing two polypeptide chains. So this is quaternary structure, and this is a folded structure of protein. Interlinked via disulfide bond or disulfide acid. This is tertiary. Accordingly, proteins are termed as dimeric, monomeric, trimeric, or oligomeric. If one chain of amino acid is there, the protein structure or protein unit is known as monomeric. If there are two amino acid chain, they are termed as dimeric. Three chains are termed as trimeric, and if there are more than four and up to 10 chains are present, they can be categorized under oligomeric. If the chains are similar or have the same amino acid sequence, these are called homomeric, or chains are called as heteromeric if they contain different amino acid sequence. Protein, there are also another classification of proteins, and they have also been put into two major groups or classes. One is fibrous and other is globular. Fibrous protein having polypeptide chains arranged in a long strand or sheets, while globular proteins having polypeptide chains folded into spherical or globular shape. So these are two main other classes or two other groups or classifications of protein based on their structure or folding. The two groups are structurally distinct. Fibrous protein usually consists largely a single type of secondary structure, while globular proteins often contain several types of secondary structures like alpha caroteins are predominantly alpha helix, whereas silk proteins are beta sheet. The two groups differ functionally in that the structures that provide structure, support, shape, and external protection to the vertebrates are made of fibrous protein, whereas most enzymes and regulatory proteins are globular proteins. There are some other functions of proteins. Proteins carry out most diverse and possibly the largest volumes of cellular functions. Some of the key functions are summarized as below. The first one is biocatalysis. Almost all the biological reactions are catalyzed by enzymes. These are substrate specific and carry out reactions at very high rates under mild physiological conditions. Several thousand enzymes have been identified to date, and all enzymes are basically proteins. Membranes are constitute of the lipoprotein, and some proteins are integral part of membrane. Receptor found on the membranes are also protein in nature. Transport and storage proteins. Small molecules are often carried by proteins in the physiological settings. For example, hemoglobin is responsible for the transport of oxygen and tissues. Muscles are made up of proteins and their contraction is done by the actin and myosin protein. Support is also provided by protein like skin and bones are strengthened by the protein collagen. Antibodies of immune systems are protein in structure. Many of the hormones and growth factors such as insulin and thyroid stimulating hormone TH are protein. So this is the brief introduction about protein classification and functions of proteins. Thank you.